I'm writing the live presentation high, so let's all learn about why the shape of your prostate matters. Even if you may not have a prostate, this might be applicable to you for some very odd, obscure niche reason. So, welcome to my mostly informative lab presentation on nuclear morphology and how it drives disease. On the top left, we have prostate cancer. On the top right, we have more prostate cancer. On the bottom middle, we also have prostate cancer, and all the cells in the background, shocker, it's prostate cancer. So let's get into it. Of course, we must start with disclaimers. One, this is mostly about my research, so a lot of this is preliminary. Science is meant to be research agreed upon and refuted upon. So if you happen to be an expert on nuclear morphology or cell biology in general, please tell me your hypothesis on anything I'm saying, and please tell me whether or not you believe it, because sometimes I find it hard to believe my own research. I'm also not a doctor. It's very important to delineate science versus doctor. I'm trying to figure out what's happening. I'm not trying to diagnose you with cancer. If you feel like there's something wrong with your body, please go to a real doctor and don't listen to a random person on the internet. I'm also not curing cancer. This is a very common misconception with people who do research and scientists in general. This is the reality of research. A lot of people aren't curing anything. I'm much more in the foundational knowledge realm of research, so maybe all of my research I can pass on to someone who's more well equipped in maybe some medical research, and maybe they can then cure cancer. I also wonder if people care. Do people still care about science in our current administration where woke is uncool, science is uncool? Guys, I promise science is cool. Please don't click off this video. Let's also try to use layman's terms. I'm sure a lot of non-scientists and scientists, aka me and then my sister, will be listening to this. So I want to make this a very pleasant and understandable listening experience for anyone. On the right, I have rotavirus. These green little guys, those are replication chambers for the virus in the cells. This is not what I'm talking about. I just thought this picture was cool and I did this research 2 years ago. So maybe in a future video I can talk about that. This is also a representation of me in lab just trying my best. I'm not blonde and I'm not a stout man, but I am Asian, so maybe something about the stereotypical skin color is resonating with me. I don't know. Anyway, let's move on. Okay, in my previous heated rivalry video, which for some reason talks about prostate cancer, I talk about politically correct prostate cancer. I have to amend my sins. I need to talk about even more politically correct prostate cancer. Guys, you don't necessarily need fingers up your ass to figure out if you have prostate cancer. That is a digital rectal exam. There's also the PSA blood test. This basically just tests for an antigen in your blood. If you have high levels of this antigen, it's highly associated with prostate cancer. If you have low normal levels, you're probably fine. The PSA is actually more sensitive than the digital rectal exam, but these tests can be used in combination to help diagnose you. I also talked about how you should get tested once a year at age 50. So the age of testing actually depends on your risk. If you are a high risk individual, then your testing should actually start around age 40. High risk individuals may include black men if you have a family history of prostate cancer or if you have certain genetic mutations. Again, you know your body the best, so this is just a very baseline around the age 40 you should start getting tested. But if someone close to you recently had prostate cancer or got diagnosed with prostate cancer, you should probably also get tested. The moral of the story is to just take care of your own body. Again, you know your body the best, and you are going to have to be your best advocate because the US healthcare system is bullshit. There's also a few things that can affect your PSA test results, and I kind of want to talk about them because they're funny. One of them is a UTI, not funny. Another one of them is vigorous exercise, kind of funny. Another one is some medications can affect your results, as well as if you had anal sex recently. So just gotta check off all these things before you get tested. Now, getting into my actual research, I deal a lot with the nucleus. So, what even is the nucleus? According to me, the nucleus is the powerhouse of the cell. Others may disagree with me. You might say it's the mitochondria, but the mitochondria and the nucleus actually have a lot in common. They both have DNA. They actually communicate with each other through something called mitochondrial retrograde signaling. None of this is layman terms. I'm just throwing words at the wall, so you can ignore all of that. What you should know about the nucleus is that it stores your DNA. Your DNA is your genetic information. Your genetic information is what makes you you, and you're awesome. So by some transitive property, DNA is also awesome. It's also pretty important to keep your DNA safe. When your DNA has a mutation, that's when your DNA gets messed up. That can lead to cancer, and we know cancer is bad, so DNA mutations, by the same transitive property, are pretty bad. It's also important to note that nuclei are only in eukaryotic cells. So, multicellular organisms are normally eukaryotic. Let's think plants, let's think fungi, let's think animals like us. There's also some smaller guys that are also eukaryotic and are single cellular, for example, protists. But just know that most of the cells in your body that you are living in right now have a nucleus as its control center. It has all the DNA that is going to make all the proteins in your body. Sounds pretty important. When it comes to the nucleus, the shape matters a lot, and actually the size does too. There are two main structural components of the nucleus. First is what the heck? First is chromatin. Chromatin is DNA and proteins bound up into this spaghetti mixture. On the diagram on the right, we can see all of the purple little squigglies in the nucleus. That's chromatin. The other component is lamins. We have three specific lamin proteins, lamin A, B, and C. This creates the meshwork surrounding all the chromatin. In the diagram, we can see that the green X's surrounding all the chromatin, that is our nuclear lamina. So these two in combination help resist any forces pushing against the nucleus to maintain its shape. The lamin is like a little net, and the chromatin is the spaghetti in the net, and this together is helping keep that nice round three-dimensional shape. There's also something called actin pushing on your nucleus. Actin is the green little guys in the diagram in your cytoplasm. This is going to be an antagonistic force on the nucleus pushing down, and the lamins and the chromatins in combination is resisting this force. So on the bottom, I took two images, one of chromatin and one of lamin. The one on the left is chromatin. Chromatin can actually localize around the edge of the nucleus. This helps maintain its shape. Specifically, this is heterochromatin, H3K9 trimethyl, if you want to look that up. For some reason, whenever I see edge chromatin, it reminds me of that meme. What is it called? None pizza, left beef. Just every time I see chromatin localized around the periphery, I think none pizza, left beef. I don't know why, but that's how I think about it. And then on the right, we have a picture of lamins. I think this is lamin B1. Lamins are strong too. Yeah, I thought these were just some nice images. So now that we know that chromatin and lamins are helping stay or is helping our nucleus stay in the correct shape, what happens if your nucleus is a little wonky? What if your chromatin and lamins can't actually resist all the pressure that is getting pushed down on it? So what we know is that abnormal nuclear morphology is associated with many human diseases, for example, cancer, progeria, and more. It's also used in diagnostics for breast cancer, prostate cancer, and other cancers. Prostate cancer, that's what I'm talking about. Breast cancer, we have a breast cancer cell line that someone in our lab is currently studying, which is also very cool. A lot of the cells we also use are mouse embryonic fibroblasts, which are epithelial cells. That's the cell that you're looking at right now in this picture. So in the cell on the left, very coquette, very nice, uniform, round cell. Not a cell, round nucleus. That is the ideal shape for a nucleus. On the right, this one's a little ugly. This one's a little oblong. But in particular, what we should be looking at, this white arrow is pointing to this little nuclear protrusion. That shit is ugly. We know that this is abnormal, that this is bad, and this can actually make your nucleus explode. We'll see that on the next slide. But just know that these nuclear protrusions are associated with disease. For example, in my previous heated rivalry video, I talked about Gleason scores. A higher Gleason score means more severe prostate cancer. And actually, the more nuclear protrusions you see in your prostate cancer cells, the higher the disease is. So I'm trying to figure out why this shit is happening. Why are these little nuclear protrusions appearing? So, dead ass, the nucleus can explode from that little nuclear protrusion. I have two images of the nucleus. The one on the left is taking at time equals 0, and we see something a little suspicious, something sinister on the bottom of that nucleus. That is a nuclear protrusion. We're looking at nuclear localization sequence, NLS-GFP. This is a live cell. So all of the green, the bright green, that's all of the proteins in the nucleus. This is moments before disaster. Fast forward, time equals 2 minutes. On the right, we see that the nuclear protrusion's actually gone, and that the bright green is actually a lot dimmer, and it's brighter around the cytoplasm, the area surrounding the nucleus. This is because the nucleus exploded. That little nuclear protrusion gave way, it exploded, and all of the green protein is flowing out of the nucleus. That cannot be good for your nucleus, and it definitely isn't because that can cause cellular dysfunction independent of increased DNA damage. Not good. We do not want our nuclei to explode, but isn't it really pretty that we can visualize the nucleus 2 minutes before its explosion? It's pretty cool. And I also have a movie that I cannot play. Let's try to play it. Quick cut because I cannot play the movie. Okay, it's kind of playing. Fuck, it's not playing. Let me try again. Let me try again. Oh, okay, it's playing. It's playing. Well, we'll watch it here. So I took a time lapse of this nucleus. We can see that over time, the nucleus will become a lot dimmer. Every time it became dimmer, that's actually it exploding. So it explodes over and over again. Yeah, that can't be good for you. That certainly cannot be good for you. But isn't this, it's a cool movie. It's a cool movie to watch. And that's all due to that stupid little nuclear protrusion that I'm trying to figure out. Why the hell are you even there? Next slide. Thank you. Okay. So the protrusion itself actually causes DNA damage. The explosion doesn't cause DNA damage, but as long as you have that little protrusion, your nucleus is messed up. So this is the same two nuclei we saw before. The nucleus on the left, there's not really a lot of bright spots when we're looking at the 53BP1. The image on the right, there's not a lot of bright white spots. It's a very much uniform, even spread of white. But on the right, we can see a bunch of little dots, a bunch of bright little spots. This is actually 53BP1, which marks double strand breaks. So basically, all of these little bright spots is your nucleus trying to fix all the broken fragments of DNA. Yeah, that's not good. You do not want that to be happening in your cells. Do I know why this is happening? Probably physics causing this, probably something with the nuclear protrusion offsetting all the chromatin, and then things are breaking. I don't know. My specific hypothesis is that the cell's freaking out, like when you wake up and you see a pimple on your face and you try to put a pimple patch and a bunch of products on it to get it to decrease before it overruns your face, but then all the products that you put on is actually overcompensation, and then you actually break out way more than you actually would have intended to had you not done anything about that single pimple. That's my hypothesis. The cell's freaking out. It's like, oh my god, I have a pimple on my face, and then it's like it's not that deep. It's not that deep. Anyway, so a lot of the cells we were looking at before are mouse embryonic fibroblasts. I've been looking a lot at 3 prostate cancer cell lines because I've been sequestered to that hellscape. For some reason, these bitches don't explode. So one of the cell lines explode. The other two, I guess they're just hardy. I guess they just know something that I don't. They know something that all of our other cells don't because they just don't freaking explode. I literally don't know why. If we look at the images on the left, these RD145 cells, they're really wrinkly. I don't know why. I'm just kidding. I do know why. It's my fault. I just didn't do a good job on the experiment. But I really like how they look because you can kind of see all the wrinkles. The image on the right is the lamina. You can see all the wrinkles and how intricate the lamina is. And it's crazy how, like, cells are in us. Maybe not specifically these prostate cancer cells, but we're made of cells. And then my job is to look at cells under a microscope. Can I look at myself? Like, am I gonna see something strange in the cell? My biggest fear one day is that I'm gonna look at a bunch of cells under the microscope and they're gonna arrange themselves in such a way that a subliminal message is being sent. Like, they create letters in specific orders and then I have to decode them and then it's actually giving me a secret to the universe. That's my biggest fear because I think I might actually go into psychosis. Um, Okay, that's an aside. On the bottom, I made a 3D rendering of one of the cells above. So this is a prostate cancer cell, 3D rendered. Purple is chromatin, blue and white are lamins. I think this is just a nice representation showing that your chromatin's on the inside and then your lamins kind of hug your chromatin on the outside. This is a bad 3D rendering. I was just bored, but this is giving you the general idea that your nuclear lamina envelops your chromatin. Okay, the age-old question. Why do these nuclear protrusions form? Why do our cells make these and why do our cells hate these? I don't know. I don't know shit. This is the crux of research. We know this phenomenon is happening. We've studied this phenomenon for so long. Why don't I know why it's happening? We do have an idea though. There must be some sort of combination between a gap in the nuclear lamina, external force pushing down on the nucleus, and internal movement within the nucleus. These 3 things create a perfect storm that creates a little nuclear protrusion that messes up your entire cell and then gives you cancer. How am I going to test that this is happening? It's three separate things, and all these three separate things have probably 100 separate things affecting them. So it's going to be really hard, but that's the general idea for now. I must do super resolution first to see the Lamin gap because the normal limit of resolution for a microscope is way bigger than the Lamin gap will ever be. So maybe if I have time somehow in the day, I can just do that. I did learn how to do STED imaging, which is a form of super resolution microscopy. So I guess when I do have time, I'll do it. But right now, definitely not. There's also way more things that we could talk about regarding nuclear morphology, such as how does transcription affect nuclear morphology? What about the cell cycle? Mitosis, artificial force, measurements, chromatin Lamin linkers, or confluency. Let's not talk about these. Sometimes when you get too deep into a topic, you start spinning in circles, going down a rabbit hole. Let's just keep this very surface level. As long as the shape of your nucleus is good, you're good. So, in conclusion, cells slash nuclei come in all shapes and sizes, but some shapes are better than others. Not very woke of me, but you want your nuclei to look a certain way. If they don't look a certain way, then we are in trouble. Science is also cool, but it's also a prison. Like, I wish I was a rock. I wish I could just wake up, see the sunrise as a rock, sit on the pavement, and then watch the sunset. But instead, I have to wake up, see the sunrise, go to work, look at cells, come home from work while I'm driving. I see the sunset, and then I have to eat dinner and then go to sleep. It's a very cyclical, sick thing, but at least science is cool. At least at the end of the day, my pretty little cells are pretty cool. So stay healthy, or at least try to. Cancer is a random event. Some people are more predisposed to cancer than others. The best you can do is to keep healthy, listen to your body, and advocate for yourself. If you feel like something's wrong, then go see a doctor. And yeah, take care of your body because you only got one of them. You don't want to mess up the one body you have? I do really need to transition into writing Boys Kiss. I feel like I will reach self-actualization if I can monetize me writing Boys Kiss. As much as I love science and as much as I think science is cool, I think boys kissing is also cool. So I really gotta lean into that. Follow my Twitch, link in bio. Shameless plug. I don't even really stream. I just sometimes when I come home from work, I'll play a game of two of Valorant with my friend, and then I'll like lose my mind. And it's very cathartic, to lose your mind just for a little bit after a long day. I'm also never buying a house. I don't think anyone in our generation is buying a freaking house. I just looked in the town that I work in, the average cost of housing is like $100,000 more than the national average cost. And I'm like, I need to rob a rich person. Or I need to rob a rich person or I need to marry rich. I feel like those are the only two options at this point. If you guys know anyone, hit me up. And yeah, at the end of the day, I just want to be like Squidward on the right. I just want to spread the woke agenda. I just want people to realize that science is cool. Support your local scientist. Support your local library. Support your local gay person. I don't know what I'm saying. I think I need to end this video. Science is cool, guys. That's all I can say.